Hello, children. Today's topic of discussion is operon. We will be looking into the definition of operon and different parts of operon, and then we go down to understand what is lac operon. So let us see what is operon first. If we go by the definition of an operon, it goes like this: a stretch of DNA consisting of a cluster of gene that acts as a single transcription unit synthesizing a single RNA molecule. That means that it will have different genes, cluster of genes in the DNA, which ultimately will help in synthesizing an RNA molecule, to be more specific, messenger RNA molecule. And this messenger RNA molecule will help in the translation of a protein or different kinds of components. It's going to help in the formation. Basically, when we talk about the operon concept, this is seen mostly in prokaryotes. But very few organisms like C. elegans and fruit fly Drosophila melanogester, they have this black operon or this operon functioning in their body. Otherwise, most of the operons are found in prokaryotic organisms. When we look into the parts of an operon, I'm just moving this slide. I'll come back to the diagrammatic part when I'll be explaining. The components of an operon, there are some majorly 4 components and one more component which we will be discussing a little later, which comprises of an operon. The first one is the structural gene. I'm reverting back the slide. Structural gene means set of genes. Here you can see X, Y, Z. These are set of genes which are responsible for the formation, for the transcription of a messenger RNA, which ultimately will help in the formation of an enzyme. So they are the group of genes which have to be coded into messenger RNA. Next one is promoter. Here promoter you can see in the diagram, which is denoted as P. This promoter is a site where the RNA polymerase is going to join to the promoter so that the transcription is possible. So promoter helps in the giving a binding site to the RNA polymerase for the transcription. Next one, it is operator. Let's revert back the slide, which is denoted here by O. Basically, this operator region is present adjacent to the promoter region, and this operator region has a tendency of binding to a protein which we call it as a repressor protein. And if the operator binds to the repressor protein, it will not allow the promoter to move ahead for the transcription. So it will become a kind of an hindrance in the transcription. The third and the next one is the regulatory gene. Now regulatory gene, you can see in the diagram, is denoted by I. This regulatory gene is a set of gene which goes for the transcription of a type of messenger RNA which will result into synthesis or the formation of repressor protein. Now this repressor protein has a capacity to stop the whole functioning of the operon. So that is synthesized by the zone which in the diagram is represented as I, and that is called as regulatory gene. To stop the regulatory gene is going to synthesize what? It's going to synthesize a messenger RNA, and messenger RNA is going to synthesize what? It is going to synthesize a repressor protein. Now to stop this repressor protein from stopping the whole operon, there is one more component which is called as inducer. I'm just moving this slide. This inducer is a substrate which will go and bind to the repressor protein, thus preventing this repressor protein from stopping the functioning of the whole of the operon. This is a general idea of any of the operon, and these 4 parts are present with all the operons. One more part which is not actually mentioned in the diagram, it is called as CAP binding site. What is CAP binding site? It is called, the full form of it is catabolite activator protein. This CAP, catabolite activator protein site, is present next to the regulator gene. You can see it is in the gray zone. This CMP binding site helps in the moving of the transcription forward. That means it helps the promoter to go and start the transcription. But this CMP site gets functional only when in the medium the glucose content is very less or there is no glucose content. If glucose is more, this CMP site is switched off. So CMP binding site and this repressor protein, these are two types of proteins which are governing the functioning of the operon. Repressor protein is going to stop the operon functioning, whereas CMP binding site, which will result into the formation of CMP protein, will help in the moving of the operon concept, operon transcription. This is a general idea. Let us now go through the definition of an operon. What is an operon? It is a unit of a prokaryotic gene expression which includes coordinately regulated, that is the structural gene, and control elements which are recognized by the regulatory gene product. Then what are the components of the operon? First, the structural gene, fragment of DNA which transcribes messenger RNA for the polypeptide synthesis. Second, promoter, sequence of DNA where RNA polymerase binds and initiates the transcription of the structural genes, and they are called as promoters. Third, operator, sequence of DNA adjacent to the promoter where a specific repressor protein binds and is called as operator. Regulatory gene, the gene that codes for the repressor protein that binds to the operator and suppresses the activity. As a result, transcription will be switched off. Inducer, substrate that prevents the repressor from binding the operator and is called as inducer. As a result, transcription is switched on, and it is a chemical of diverse nature, metabolite, hormone, substrate, etc. Now, this is a general idea of an operon. Now, let us go into the lac operon that will make our concept more clear. Now, see, this is the diagram of a lac operon. I am just reverting a little bit more so that you can understand. This is the diagram of a lac operon. As usual, I'm just reading it out. I is the zone which is going to be called as the regulatory gene, and this I will help in the synthesizing of a messenger RNA, which you can see, which will ultimately go for the formation of a repressor, which is also seen in the slide. This repressor is a type of protein. Next to the I, there is P. P is the promoter zone where the RNA polymerase is going to join, and it will help in the movement or help in the transcription. Next to the promoter is operator. Operator also helps in the movement of the transcription. After that, there are three genetic genes are present: Z, Y and A, Z, Y and A as it is called. Z is the area, I am reverting back the slide for your better understanding. This LAC-Z will help in the synthesizing of an enzyme called as beta-galactosidase. This beta-galactosidase is responsible for breaking down lactose into galactose and glucose. So it helps the lactose compound to act as a energy producer in case of prokaryotes. So beta-galactosidase helps in breaking down of the lactose, and from where it is synthesized with the help of a gene called as LAC-Z. Then LAC-Y. LAC-Y helps in the synthesizing or the transcription of a enzyme called as permease. What's the function of permease? Permease will allow the movement of lactose from the medium inside the bacteria. Bacteria will be present in a you can say nutritive medium. So bacteria will be having its own cell membrane. So lactose is present. We add a lactose in the medium. We cannot inject the lactose inside the bacteria. So this permease will make the cell membrane of the bacteria more permeable for the lactose to be absorbed inside. So lactose, it helps in the transferring, or you can say that it helps in the permitting of the lactose inside the bacterial cell. So that is coded, or that is transcribed with the help of enzyme sequence called as LAC-Y. Last one, LAC-A, it helps in the synthesizing of a protein which is called as enzyme which is called as trans-acetylase. Now this trans-acetylase helps in the lactose to become lactose into active form. So that is synthesized with the help of LAC-A. So this is actually the operon. This is the, we can say that a kind of a master diagram of a lac operon. What it consists of, I'm repeating again, it will consist of some genes which are called as regulatory genes. So we can call it as I, which will synthesize a repressor protein. Then the promoter zone, which will help in the binding site for the RNA polymerase. Then comes the operator zone, which will help in the transcription. And then comes the LAC-Z, LAC-Y and LAC-A. LAC-Z will help in the formation of the enzyme beta galactosidase. LAC-Y helps in the formation of we call it as permease, and LAC-A helps in the formation of an enzyme called as trans-acetylase. So this is the operon. Now what happens in this operon? Sometimes this operon is called as switched on and sometimes it is called as switched off. Now why that is said like that? For that, let us go down to the diagram again. Now imagine a situation, you have a solution of lactose and some bacteria you put inside that lactose solution. Now what will happen? The bacteria will want the lactose to be used as an energy source. So what will happen once the lactose is present in the medium? I'm moving ahead with the slide. Once the lactose is present, this I, you can see in the diagram, this I zone, which is called as regulatory zone, will definitely synthesize messenger RNA, and as a result of synthesizing of messenger RNA, it will result into formation of repressor protein. Now this repressor protein has a tendency of going to the operator and joining with it, thus hindering the process of the transcription. But when the lactose is present, which will act as an inducer, this lactose will go and join with the repressor protein, you can see in the diagram, and it will make a inducer repressor complex, which is the structure of this inducer repressor complex will be so different that it is not able to join with the operator. Thus, this repressor is not going to stop the operator, and in the presence of promoter and RNA polymerase, the transcription of Z, Y and A genetic code, the structural gene, is possible. And ultimately what will happen when RNA polymerase is going to transcript Z, Y and A, it will make a messenger RNA, and we all know that messenger RNA in case of prokaryote, they are polycistronic in nature. That means one type of structural gene can make all kinds of enzymes. So Z, Y and A will go for the formation of a messenger RNA, and with the help of that messenger RNA, beta galactosidase, permease and trans-acetylase enzyme will be formed, and this condition is called as operon switched on. But if in case this lactose is not present, once the lactose gets used up, the quantity of the lactose will keep on reducing in the medium. So there will be a point where lactose will be not present, it will be finished off. Now that what will happen at that time? I'm reverting back the slide where the lactose is not present. So same thing will happen. I, that is the regulatory gene, will synthesize messenger RNA. Messenger RNA will synthesize a repressor protein. But here, now no one is there to stop this repressor protein, which has a tendency to go and bind with the operator. So, repressor protein will go and bind with the operator. Ultimately, the promoter, which is having the binding site of RNA polymerase, can bind with RNA polymerase, but as the repressor has stopped the way of this RNA polymerase to continue the transcription, this genetic codes Z, Y, and A cannot be transcribed into messenger RNA. So such condition is called as operon switched off. That means the repressor has binded to the operator, thus the operator becomes non-functional, and promoter, which is having the RNA polymerase enzyme which is responsible for the transcription of the Z, Y, and A, cannot move ahead. This repressor acts as a, you can say, a kind of a barrier. It stops the movement of the RNA polymerase. Thus the operon is switched off. Remember, operon can be switched on when we put an inducer in the form of lactose. Thus, the repressor will be binded to it and it will not go and attach itself to the operator, and the whole of the operon will be functional. So, when we say once the operon is controlled by the repressor in this way, we say that repressor activate is negatively influencing the operon. But there were, if you remember in the initial part of this lecture, I talked about a CMP site which is present adjacent to I. Here it is not shown here. That is the regulatory gene. This CMP site helps in the movement of the transcription forward. So once it helps in the movement of the transcription forward, it influences this operon positively. So when the CMP site, which will form a protein called as CAP, that is cap protein, and it will influence the operon in moving ahead, moving for synthesizing. This type of work is called as positive movement, or it is called as positive influence. Whereas when we talk about the repressor goes and binds to the operator and does not allow the promoter to move ahead for the transcription. So this is called as the negative impact on the operon. I'm now reading the slides to make it a little bit more clear. Lac operon, this we have already gone through it. Lac operon, the lac Z, lac Y, lac A genes are transcribed from lac transcription unit under a single promoter. Coded enzymes are required for the use of lactose as a carbon source. Lac I gene product, lac repressor, is expressed from a separate transcription unit upstream. Three structural genes Z, Y, and A transcript in the polycistronic messenger RNA. That's what I said. Z, Y, and A are the genetic regions which will go for the synthesizing of a single messenger RNA, which is polycistronic in nature, because this thing happens in prokaryote, and this messenger RNA is capable of synthesizing beta galactosidase, permease, and transacetylase. When lactose is absent, I gene regulates and produces a repressor and repressor messenger RNA, which translates into a repressor protein. The repressor protein binds to the operator's region of the operon, and as a result will prevent the RNA polymerase to bind to the operon. Now the operon is switched off. This is how the things will happen. When lactose is present, lactose acts as an inducer which binds to the repressor and forms an inactive repressor. That means it will make the repressor structurally so different that it's not going to attach to the operator. The repressor fails to bind to the operator's region, and RNA polymerase binds to the operator and transcripts messenger RNA. Lac messenger RNA is polycistronic, produces three enzymes, beta galactosidase, permease, and transacetylase. Thus, the lac operon is switched on. You can see the diagram here. Again, for the better understanding, one video I am attaching to it. Let's have a look at it.
And they shared the Nobel Prize in for their tremendous work. What is Operon? Operon is a functioning unit of DNA containing cluster of genes under the control of a single promoter. Operon concept explains the metabolism of lactose in prokaryotes. It consists of various genes help in lactose metabolism by producing different enzymes. Before moving to the concept, let's understand the terminology. P indicates promoter, O indicates operator, I indicates the gene, regulator gene. The structural genes called LAC-Z-Y-A. The gene I is called regulator gene, which codes for the repressor of the Operon. Repressor is a small protein formed by regulator gene, which binds to the operator and block the passage of RNA polymerase towards structural genes. Once the repressor binds to the operator, the Operon becomes switched off. In the presence of inducer, nothing but lactose, which attaches to the repressor and changes the shape of operator binding site. Then operator becomes free. As soon as the operator gene becomes free, RNA polymerase is recognized by the promoter gene. Then RNA polymerase passes over the operator gene and then reaches the area of structural genes. Then Operon is switched on. Structural genes take part in synthesis of polypeptides, proteins, enzymes through the formation of specific mRNA. This mRNA is known to be polycistronic, which produces all three enzymes called beta-galactosidase, permease, and transacetylase. An enzyme beta-galactosidase is primarily responsible for the hydrolysis of the disaccharide. The enzyme permease increases the permeability of the cell to beta-galactosidase. Enzyme transacetylase catalyzes the transfer of acetyl group from one molecule to the another.